So today in IST 455 we're going to talk about drug target identification. So drug target identification is a business decision, and there's a lot of factors involved in selecting the appropriate target. A pharmaceutical company has to have a good understanding of the disease in order to identify the correct pathway to interfere with or to turn on for the drugs to be effective. They have to identify an unmet medical need, because it is difficult, if there isn't a need for this drug, to create demand for it. You can create demand for a pink iPhone, but you can't really create easily demand for a medical problem. It has to be a real problem. So they also have to look at what kind of current therapy is available for the medical need. So the drug that the pharmaceutical company is developing has to work better than the current therapies. So they may identify that peripheral nerve pain in diabetes is a big problem, but there's some other drugs out there on the market that treat peripheral nerve pain, and so if you're trying to develop a new drug for this application, it needs to work better than the current therapies. Less side effects, more effective. So they also need to find out that the marketplace is big enough to pay for the development time for the new drug. There has to be a big enough marketplace. If a drug is for a smaller marketplace, it may be a good drug if there's no competition. It may not have to be the biggest marketplace as long as the marketplace really wants that drug. They need to look at their patent portfolio. Sometimes a pharmaceutical company will have part of the patent portfolio required to develop a technology, but not all of it. And so they have to then look and see if they can acquire some aspects of the patents that are needed in order to bring this drug onto the marketplace. They have to look at how complicated is this drug going to be to bring out onto the marketplace when the FDA looks at it. So if there's, for example, a high side effect profile for this drug, it may not be a good choice because the FDA is going to demand evidence that it's really an effective drug to make up for the risk of the side effects. And it should be something that falls within the company's core competency in terms of manufacturing. So if a company is used to making small molecule chemical drugs, and all of a sudden they are called upon to develop a monoclonal antibody therapy, they may have to acquire a partner or work with another company because that's outside their core company competency. So in order to identify what's needed in drug discovery, first step is to identify and define medical needs. And then you have to identify the disease mechanisms. You have to identify and validate the targets that are involved in the disease process. So for example, a receptor, you may be able to identify a molecule that blocks the receptor and prevents the disease from continuing. Or you may target a receptor that would specifically bring in a toxin and knock off a cancer cell. So then the next step is to look for lead compounds that interact with the targets. So the lead compounds are going to be compounds that are the most effective. So there's a lot of compounds that might affect that receptor, but you need the ones with high binding efficiency. Then once you've identified a compound that looks good, you can tweak that compound and modify it to generate potential drug molecules that might have properties that are effective. And then once you've got those lead compounds identified, you're going to develop the drug and do preclinical studies. So the process is usually to start out with an in vitro assay, maybe working on cells in culture, and then move towards an in vivo assay using animal models and eventually human clinical testing. So there's a couple of different ways that pharmaceutical companies identify the lead compound through a process called drug screening. One way is could be called the irrational approach or the brute force approach. So the pharmaceutical company may synthesize thousands and thousands of drug candidates. They just go through and make many, many compounds, one after the other. And then they would take those compounds and screen them in an assay with a carefully chosen target in an assay that is good. So what they want then, if they have 50,000 target molecules, then is to run them through assay after assay after assay. And out of those 50,000 target molecules, maybe only 50 or 5 would be good lead compounds. One way that pharmaceutical companies work is sometimes they'll go through and buy all the drug candidates from smaller companies. Or if you're a chemist at a university, you might make a series of compounds and then license them to a pharmaceutical company. A second approach is called the rational approach. So that the idea is that you will crystallize the receptor, for example, and then analyze its the amino acids that are in the binding site for the receptor, and then design a drug that would fit that target, and then screen the compounds to show that it's effective. And so this is the strategy of companies like Virtus Pharmaceuticals. They're focusing on rational drug design. So one of the ways that drug design and drug discovery work then is by developing a really useful in vitro assay. It could be using a cell culture, or it could be using cellular extracts and an enzyme assay, and then look for the drugs and the drug lead compounds that are going to be effective in that in vitro assay. So if you've identified your therapeutic target correctly, you're going to be able to design a specific in vitro assay to work on that therapeutic target. So, and again, it's very important to identify these unmet clinical needs, because these are the drugs that are going to be covered most effectively by insurance. The biological action of the in vitro assay then will indicate that it could potentially, the new drug may have therapeutic utility. So some of the drug research areas then could be broadly based, and then within that broad drug research area, you might target a particular disease state. So for example, in the central nervous system or neuroscience, you might target depression. In the cardiovascular area, you might target hypertension or cholesterol drugs. In gastrointestinal diseases, you might target peptic ulcer or heartburn. In infection, you might target HIV or a specific other disease like MRSA. In cancer, you might target a specific type of cancer like breast cancer. In inflammation or allergy, you might target an autoimmune disease like arthritis. In a respiratory disease, you might target asthma. For metabolic diseases, you might target diabetes. So in those broad areas, then you still want to move in and target a specific drug. So one of the big powerful tools of drug discovery has been the Human Genome Project, which was completed around 2001. The draft sequence of the human genome was completed, and since then we've been focusing a lot of attention on trying to identify drug targets. So the process then of the fundamental genetic process is that the DNA molecule is transcribed into a messenger RNA, and then that messenger RNA is translated into a protein. So if you want to affect that protein, turn it on and turn it off, you might be able to target it at the level of transcription, you might be able to target it at the level of translation, or you might be able to target the protein itself by affecting its active site or by leading it to changes in its degradation. So one of the tools that has been developed in order to identify novel ways to rapidly screen in an in vitro assay have been microarrays. So the idea of a microarray is that what if you could conduct hundreds, thousands, or even millions of experiments in parallel with very few reagents, using no test tubes, just work in very small amounts, and then have a very sensitive assay that you could detect binding. And so the microarrays then are a way of doing this. They combine genomics, which is the study of all the genes in the genome, with experiments, and eventually can be used for diagnostics. So the original microarray was the idea of using a DNA microarray. So the DNA microarray was a good method for identifying targets for differential expression of genes in different tissues. So the idea of the microarray is that you will have a glass slide or a silicon wafer, and on it you're going to put, on these little spots, individual gene products. And then you're going to create probes. So the probes in this case are genes that are differentially expressed in two different tissues. So you can have a normal tissue or you can have a tissue from a tumor. And so the tumor is presumably making a different group of genes and gene products than the normal tissue, and that's why it's a tumor. So what you would do then is purify the RNA that's made in the tumor tissue and the normal tissue, and then make a DNA copy of the RNA using a process called reverse transcriptase. And so what you would do is incorporate a label, a fluorescent label, that would be different in the tumor messenger RNAs turned into cDNAs than in the normal cells. So in this tumor tissue, you would make a fluorescent red probe, and in this normal tissue, you would make a fluorescent green probe. So what you can do then is hybridize these probe RNAs to the different DNA spots that are on the microarray. So this is a large population of messages, and when they hybridize to a specific gene, it's going to light up. So for example, if the tumor gene was expressed, it would give you a red spot. If the normal gene were expressed, it would give you a green spot. So how do you get the yellow spots? The yellow spots occur when you've got both the green and the red hybridizing to the spots on the microarray. So some tissues, some genes are important in all tissues. So those would be the yellow genes. Sometimes we refer to those as housekeeping genes. So if you're trying to inactivate the tumor gene, you might want to turn off those genes that are turned on in the tumor cells. So the green are normal, the red are abnormal, the yellow are both. And so it's important then to go back and identify what are the known DNA sequences. So scientists have looked at this process of the DNA microarray, and they've decided, well, this process of spotting things could be used as a universal biochemistry platform. So they've taken proteins, for example, and then they put antibodies on the array, and then they purify the proteins from the tumor cell line and from the normal cell line, and then identify which proteins are different in which tissues. They can put peptides down and then see which peptides, which proteins in the tumor line bind to the peptides. They can put carbohydrates down and then see if there's carbohydrate binding capability that's different in the different tissues. So some of these would represent, the spots would represent different types of carbohydrates, and then the labeled proteins would bind the carbohydrates differently in the tumor versus the normal cell line. They can put lipids down and see if there's differences in lipid binding. And they can put small molecules that might interact with the receptor down and then see which proteins bind to which small molecules and if they're different in the tumor versus the normal cell. So as I said, there's a wide variety then of these different microarrays that are being developed. So some are the DNA microarrays, but they can even be modified as well. So you can use the DNA microarrays not just to hybridize to different types of messenger RNAs, but they can be used to identify genetic differences in the cells, in the tumors, versus the cells in the normal cell line. At differences that were single nucleotide polymorphisms, or in some cases they can look at chromatin binding and identify different regions that are protected by chromatin, and then identify those on the microarrays. So they can look at different chips that are able to identify microRNAs. So these are small RNAs that are expressed differently in tumor cell lines versus the normal cell lines. I mentioned the protein microarrays. In some cases they're antibodies. In other cases they're protein-protein interactions. There are tissue microarrays to look at particular binding of proteins to tissues. Cellular microarrays are sometimes referred to as transfection microarrays. So you'd make a set of cells and then transfect different genes into those cells and then see how the cells interacted. Chemical compounds and carbohydrate arrays are sometimes also used. And I mentioned the antibody microarrays. The thing that underlines the microarrays are bioinformatics. So the more you know about the genome, the more you can identify targets, and then the more you work with silicon manufacturing and targeting, you can put different molecules down onto these silicon wafers. We also need to know more about signal and image processing so that you can miniaturize the assays even more and make smaller versions of traditional molecular biology experiments. So you need to be able to develop new instrumentation, and you need to develop new software as well so that you can analyze these experiments more effectively. You need more statistics so that when you start having tens of thousands of interactions, you need good statistics so you can get significant results. So this is an example then of biological samples in 2D arrays on a membrane or a glass slide. So as you can see here, some of the spots are red, some of the spots are green, and what you're looking for is the loss of activity or the gain of activity in the target tissue that you're interested in. So another way that you can look at this is to look at the bioinformatics of metabolic pathways. So in this case, we're looking at some of the pathways that are involved in processes for apoptosis, which is programmed cell death. And apoptosis is an important process that's involved in turning off genes and turning on genes that might potentially act against tumors. So the more you understand the biological pathway, the better you can design drugs that are going to specifically target some aspect of that pathway. So Some of the targets that you might have would be phosphorylation sites that would regulate the activity of some of the receptors or some of the binding molecules along the pathway. So here you've got a serine phosphorylation site. Here you've got a tyrosine phosphorylation site. Here you've got a threonine phosphorylation site. So when this the BAD is phosphorylated, it has one activity. When it's dephosphorylated, it has a different activity and drives the cell towards apoptosis. So the phosphorylated version leads to cell survival, dephosphorylation leads to apoptosis. So if you had a cell that you wanted to drive towards apoptosis, if you could find a way to dephosphorylate this serine 136, that would be potentially a good pathway to interfere with. So once you've selected a target, you really need to figure out what the function of the target is and what disease pathway the target regulates, because some of these targets are used in a lot of different cell types. If you knew that this bad protein was more commonly expressed in an important regulatory process in cells in the pancreas, for example, you might be able to look at it in terms of pancreatic cancer as opposed to if it's expressed throughout in tissues throughout the organism. Because then if you come up with a drug that targets that pathway, you might have too many side effects. It might work on too many different, it might not be specific enough in its function. So it's important then to identify that disease pathway as an important aspect of the pathway and have it be important in a limited number of tissues. So what you're interested in is something that is known as the therapeutic index. So the therapeutic index is the amount of therapeutic agent that causes the therapeutic effect compared with the amount of that agent that causes death. So if a very small amount of therapeutic agent causes a therapeutic effect, that's beneficial because you need a large amount in order to cause death. But if the process is, if you need a large amount of therapeutic effect of agent in order to have that effect, then the problem is you're going to have a bigger side effect profile. It'll be harder to dose an individual correctly. So some of target identification is similar to what we look at when we look at enzyme and substrate binding. So you want to identify both the enzyme and the substrate and the way in which it binds. So substrates that bind are called ligands. So one technique that's been popular for many years is a technique called radioimmune ligand binding, or RAI, a radioimmune assay. And this method was developed by Rosalind Yarrow and her colleagues, and she was awarded the Nobel Prize. And so the way this works is that a known quantity of an antigen or a foreign protein is made radioactive, and then you can label it with gamma radioactive isotopes of iodine that are attached to a tyrosine. So you make a radioactive foreign protein, and then this radio-labeled antigen is mixed with a known amount of antibody to that antigen, and the two chemically bind each other. Then you add a sample of serum from a patient who contains an unknown quantity of the same antigen, and this causes the cold or unlabeled antigen from the serum to compete for binding with the radio-labeled antigen for the antibody binding site. So it competes off the radioisotope. So this is a way of identifying that that particular protein is present in the serum of a patient. So you know you can specifically identify which protein is involved using the antibody, and you can identify it by its presence in the serum because the radioactive iodine is released into the media and not bound to the antigen, which can be coupled to a membrane. So as the concentration then of the cold antigen in the serum of the individual is increased, more of it binds the antibody and displaces the radiolabeled antigen. And it reduces the ratio of antibody-bound radiolabeled antigen to free radiolabeled antigen. And the bound antigen then is separated from the unbound one, and the radioactivity of the free antigen remains in the supernatant. So for example, you can fix the protein that's bound onto a glass fiber filter and wash away all the unbound radioactivity, and then put it in a instrument that counts how much radioactivity is present. And this will give you a very exact quantity of antigen that's present in the serum that can be determined. And it can also be used to calculate binding kinetics for this process. So why would you be interested in something like this? If the binding process could be interfered with using a drug, then that would be a way of looking at an identification of new drugs. So the radioactive ligand assays then would work in a similar way. The radioactive is sensitive and specific. One problem is it does create waste, radioactive waste. Radioactive iodine is a pain to work with. So you can also label the ligand and identify the receptor that's used, and this is one of the ways that new receptors are identified, and that the ligand binding is characterized. So for example, this was used to identify the receptor for FK506, which is a useful receptor. And then once you've isolated the purified receptor, you sequence the amino acids, and then once you've identified the amino acid sequence of the protein, you can clone the gene for the receptor to get the rest of the protein. And the new sequences, new methods that are being used for for protein sequencing, need less and less protein to get some sequences. So this method, for example, of MALDI-TOF means that if you can purify a particular protein, you can identify a lot of information about it. Now you might say, well, we already have the whole genome. Why do we have to identify this specific protein that might be in the cell line of interest or the tumor of interest? And the reason is that there's a lot of post-translational modification that goes on. There's a lot of splicing of introns and differential splicing of introns, so that the proteome, all the proteins that are possible in the cell, may be difficult to determine just from analyzing the genome using bioinformatics. So it's a good idea to be able to identify specific proteins then that are important in determining the tumor or the tissue that you're seeking to develop a drug for. Doesn't need to be a tumor. So these radioligand binding assays are used to study the binding properties of the test compounds, because you put the test compound in and you can determine if it's able to compete off the radioligand from binding with the receptor. And it also helps you determine how specific is that binding. So you care about how specific it is, you care about how strong it is, and specific strong binding compounds make better drugs. So once you've identified that this target is good, you want to really validate that that target is the appropriate target in the pathway. So the targets that bind the ligands are doing so via electrostatic forces. So van der Waals forces, hydrogen binding, hydrophobic interactions, these are the forces that are binding the ligand to the target. So what are the targets then that are being identified? Enzymes are frequently the target, intracellular receptors, and cell surface receptors that are on the outer surface of cells. These are the targets that are most commonly being used for drug identification. So once you've identified a potential drug that is a target, you want to figure out what is the mechanism of action. So generally the mechanism of action is that a drug is either an antagonist that blocks a reaction or an agonist that activates a reaction. So these different mechanisms of action then are very important for understanding what's going on in the cell biologically. So the mechanism of action can be identified in an in vitro assay, and the validation of the target can be characterized in these in vitro assays. So typically when people talk about an in vitro assay, they're talking about a cell-free assay, or they're talking about a cellular assay. So if you've got a target that's an enzyme, you'd want to see that that enzyme is inactivated by the drug in cells, not just in a test tube. So once you've figured out that your target is continuing to work well with the drug that you're developing, you would then try it in an in vivo assay. So generally people start with a mouse assay and show that it works in a mouse model of the disease. So we've got a variety of different mice that we've bred to have a greater incidence of cancer. We've got a bunch of mice that are more apt to have hypertension. We have mice that are more apt to have diabetes. And so we would put our drug into these mice and see how it affects the mice. And once you show that the drug is working well in mice, you might progress to other animal models. And once it's working in other animal models, you would move towards working on it in human assays. So the active site in many enzymes is inhibited or suppressed by the presence of another molecule. And inhibitors reduce the rate of enzyme reactions. They can be used artificially as drugs. They can also be used as pesticides, and inhibitors can also be used as research tools in trying to understand basic mechanisms of action. So there's three types of inhibition that are most commonly described in enzyme activity: a competitive inhibition, non-competitive inhibition, and uncompetitive inhibition. So competitive inhibition is probably the simplest to understand. So you've got this process where you've got an active site for the enzyme and a substrate is able to bind it. When it binds, you get the enzyme substrate complex, and then the enzyme modifies the substrate in some way, either making or breaking a covalent bond of the substrate. So an inhibitor then is going to bind to the same site as the active site that the substrate binds to. And once the inhibitor binds, it generally has a better binding. It's binding, but it can't be processed. And so if it's got good kinetics then, and it's going to bind effectively, then that will block that reaction. So the substrate is no longer able to enter the active site, and if the inhibitor sticks inside the active site, you're not going to go on and have a reaction. So it basically blocks and competes for the binding of the substrate. So a competitive inhibitor has a similar structure to the normal molecule, and it can fit into that active site of the enzyme. So it's competing with the substrate and slows down the reaction. So what competitive inhibitors do is increase the Km of the enzyme, but they have no effect on the Vmax. So the rate can approach a normal rate if the substrate concentration is increased in a high enough fashion. So an example of a competitive inhibitor that's used as a drug is the sulfonamide antibacterial drugs. So if you have enough inhibitor, they're effective. If the substrate concentration is higher, then the antibacterial drug will not be as effective. So that's one downside to the competitive inhibitor process. Non-competitive inhibition works in a different way. In this case, it's the inhibitor is binding to a different area of the enzyme, and it alters, by binding, the active site of the protein or the enzyme. So here, the altered active site no longer is able to bind the substrate, and no reaction can occur. So this structural change in the enzyme then renders the active site useless. So how does this affect Vmax and Km? The non-competitive inhibitor often has a different structure than the substrate molecule, and it doesn't fit in the active site. So it's binding to some other part of the enzyme molecule, changing the shape of the whole enzyme. And it simply reduces the amount of active enzyme. It effectively acts as if it's decreasing the concentration of the enzyme. So it decreases the Vmax, but it doesn't have an effect on the Km. So uncompetitive inhibition is similar to non-competitive inhibition, except that the inhibitor can only bind the enzyme substrate complex rather than the free enzyme. So in uncompetitive inhibition, the enzyme binds the substrate, and then this additional inhibitor binds and keeps the enzyme substrate from proceeding forward with its chemical reaction. So let's talk then about some examples of how drug targets work, and how we're going to use as our example pain medication. So there's a number of very effective drugs for pain, for mild pain. Aspirin, ibuprofen, and naproxen. So these three drugs then are often used as treatments. They are often referred to as non-steroidal anti-inflammatory drugs. The maximum recommended doses of these drugs restrict the amounts in order to prevent toxicity and damage to the liver. So you can't just keep taking more and more and more of these drugs. You're limited in how much you're able to take before you get a toxic side effect. So how do these NSAIDs work? So they have several different effects, and depending on the NSAID that you're working, they may more actively work on one effect more than the other effect. So when they work as analgesics against pain, that's a central nervous system effect and a peripheral nervous system effect. So it can involve non-prostaglandin related effects. When they act as an antipyretic or anti-fever drug, they affect the central nervous system. When they act as an anti-inflammatory to reduce inflammation, they affect the prostaglandins. As analgesics, the NSAIDs are unusual because they're non-narcotic. So what then are these prostaglandins? The prostaglandins are eicosanoids. The eicosanoids are local signaling molecules made by oxygenation of 20 carbon essential fatty acids. And these prostaglandins regulate many systems in inflammation or immunity. They are messengers in the central nervous system. So how then do these things work? So let's look at the process by which there's damage to a tissue. So what happens is there's injury to the tissue and phospholipids are released, arachidonic acid is made, and then the leukotrienes result in bronchoconstriction. So then from there you've got a couple of different pathways. So there's a COX-1 pathway and a COX-2 pathway. The NSAIDs block the hydrophobic channel by which these arachidonic acids enter the PGHS synthetase 1 cyclooxygenase active site. So the cyclooxygenase are these COX enzymes. So this PGHS synthase 1 affects COX. So you can act as non-COX-2 inhibitors. Those are going to affect COX-1. The COX-2 inhibitors are a different group of inhibitors. So there's these two forms of cyclooxygenase, and some of the drugs affect them both, and some of them affect only one of the different enzymes. So the NSAIDs are non-COX-2 inhibitors, including aspirin. So they have a cytoprotective effect, and they protect the gastric mucosa and they aid in platelet aggregation. So when you've got a COX-1 inhibitor, you wind up with less of these. So the gastric mucosa is affected and the platelet aggregation is disrupted because you're turning off these cytoprotective prostaglandins by turning off this COX-1 constitutive enzyme, this constitutive pathway. So that cytoprotective effect then is a side effect. So that's why you get side effects of aspirin. It works to reduce the pain, it works to reduce problems, but it has these side effects of ulcers and side effects of platelets that don't aggregate. So they advise you, for example, not to take aspirin before surgery because it'll affect your aggregation of your platelets. That's also why they advise you to take aspirin when you have a heart attack, so that after a heart attack your platelets won't aggregate. And at that point they're less worried about your ulcers than tissue damage from aggregates. So the COX-2 pathway then is also affected by the NSAIDs and the aspirin. So, but COX-2 is an inducible enzyme pathway. So some of the inducers of the COX-2 pathway are cytokines and growth factors. So aspirin, in addition to being a COX-1 inhibitor, is a COX-2 inhibitor. And there's other specific NSAIDs that are non-COX-2 inhibitors, and then there's specific COX-2 inhibitors that are not affecting the COX-1 pathways. So the inhibitors that are specific to COX-2 are going to have a different, more favorable side effect profile because they're not going to affect your ulcers and they're not going to affect the platelets. So the inducible pathway are the inflammatory prostaglandins. So if you're inhibiting those, it's going to, those inflammatory prostaglandins, when they're inhibited, they won't, the damaged tissue won't recruit inflammatory cells, so you won't get so much inflammation. You won't sensitize the skin pain receptors, and it's also going to regulate the hypothalamic temperature control, so you're not going to have a fever. So inhibiting these inflammatory prostaglandins is going to be beneficial if you are having swelling and a fever because you've got influenza. It's going to relieve your pain, in other words. So this is a benefit. So you would prefer to have the COX-2 process inhibited rather than the non-specific effect of the COX-1. So another drug target that is interesting for people is looking at HIV. So one aspect of in the past 25 years, we've developed a large number of very different drug targets against HIV. And that's been important because the HIV virus is made by reverse transcriptase. So it readily mutates and can develop antiviral drug effects. So we've needed to define a variety of different drug targets for HIV so that we can treat individuals who have HIV with 2 or 3 different drugs and prevent the cells from becoming resistant to a single drug. So some of the aspects of the HIV drug target is that the HIV virus has a specific CD4 interaction that it works with. It attaches to the helper T cells through these CD4 proteins. So if you can block those CD4 proteins binding the HIV virus, then that's beneficial. So another aspect of the HIV that we've developed drugs against is viral RNA and reverse transcriptase. So if you can block the reverse transcriptase from making a copy of the viral RNA, then that would be very beneficial because the virus wouldn't be able to replicate. Another aspect after the virus begins to replicate is that it makes HIV proteins. So the virus makes a very long RNA that is then translated into a series of different proteins that are cleaved apart by proteases, specific proteases. So if you can block the HIV protease from cutting the proteins to their specific size, that's also a very effective drug because then the new particles aren't able to get the correct size proteins to assemble an infectious particle. So if the infectious particles can't spread, then that reduces the symptoms of the HIV. So you're trying to block it from getting into the cell in the first place. As some of it gets through, you're trying to block it from replicating its viral RNA into DNA using reverse transcriptase, and if it gets through that pathway, then you're trying to block it from making the protease so that it can make new virus particles. So all these different steps in the replication of the virus then could be targets for HIV drugs and have been developed as targets for HIV drugs. So the HIV reverse transcriptase in particular has a number of different functional aspects to it. So in this model, you can see the new DNA is being made here, and the HIV RNA is in yellow, and it's fitting into this cleft in the HIV reverse transcriptase. So one of the drugs that's been developed is an analog for the nucleotide thymidine. So thymidine is the T in the T base. So what it's got is the phosphate attached to the sugar molecule. Here's the 3' carbon in the sugar molecule, and then the nucleotide base for thymidine is over here. So AZT is one of the first drugs that was developed against HIT, azidothymidine, and it resembles a thymidine, but it's got this azido group in the 3' position of the sugar molecule. So here, instead of having a free 3' OH, you've got an azido group. So what happens then is that this analog to thymidine comes along and gets incorporated into the growing DNA molecule, and once it's incorporated, then the reverse transcriptase can no longer replicate the viral RNA into a DNA copy because there's no free 3' end to add another nucleotide base to it. So if this will act in the cells against any DNA replication machinery, but in virus-infected cells, it's most effective against HIV because HIV virus is replicating faster than other DNA. So the side effects that you're going to see are going to occur because of replication of other DNAs being affected, but it's going to effectively work on cells that are have a big virus load in them. So rapidly growing cells are going to have side effects from the AZT, but the rapid virus growth is going to be positively affected. So protease inhibitors irreversibly bind to the protease enzyme, and while they're bound, they prevent the enzyme from cutting the viral protein molecules down to their proper sizes. So this little cleft here is where a protease would bind a protease inhibitor. So the process for recognition of the HIV-infected cells by the cytotoxic T cells, here's the infected cell. It makes its viral proteins. The cytotoxic T cell then comes along, has a receptor, and recognizes the infected cell by these antigens. And so the cytotoxic T cell then is binding these. So a ligand that would bind the receptor and kick off a cascade of other reactions then would amplify the signal. So it's important then to understand the pathway that's involved in order to understand how the drug interaction can produce the side effects as well as the desired response to the drug. So these are some signal pathways that are well defined. So this is the NF-kappa B pathway. This is the JNK or junk pathway that involves cellular stress and cytokines. This is the MAPK pathway, another NF-kappa B pathway. So along these pathways then, you have all these interactions then that lead to the NF-kappa B, in this case, binding and turning on a series of genes. So what you want to do then are develop drugs that can act on a specific aspect of the pathway and either turn it on or turn it off. So I had a different version of this cell that showed how these are all working then in the cell in terms of turning on apoptosis and turning on gene cell proliferation. So in many cases what you want to do is interfere with cell proliferation if what you're trying to do is develop a drug that decreases a tumor, and you might want to turn on apoptosis specifically so that this cell, this tumor winds up committing suicide. So another drug that has been developed is recently has been a drug that can bind the active site of a protein in a specific way such that it changes the activity or reactivates a particular defective protein. So one of the problems that can occur with cystic fibrosis is that you have a defective faulty gene protein product, the CFTR, or cystic fibrosis transmembrane receptor. So this CFTR faulty gene product is this faulty protein. So cystic fibrosis is a mutation then in this CFTR